veins, we have deoxygenated, or you can also say less oxygenated blood, and that's because as the blood has gone through the capillaries, much of the oxygen has moved out of the blood to the cells where it's going to be used for cellular respiration to make ATP. So the venous flow is going to be fairly low ones. Next chapter, the respiratory, will give you specifics about the actual amounts, but we'll leave that for a little bit. Anyway, we know it's lower. And of course, it's higher in carbon dioxide. It's carbon dioxide, byproduct of cell respiration, has moved from the cells into the capillary blood and then now on into the veins. Now the veins, we have the smallest veins are called venules, and then we have the veins, and we can go to small, medium, and large veins as they're branching back, coming towards the heart. Eventually, from the systemic circuit, they go to the inferior vena cava and the superior vena cava, draining the head and neck. And those two large vessels, along with the cardiac vein, bringing blood back from the heart itself. Those three vessels, the cardiac vein, the superior vena cava, and the inferior vena cava, take this less oxygenated blood and dump it into the right atrium, which dumps it into the right ventricle, which then goes to the pulmonary circuit. So the veins, as they get bigger, can have some smooth muscle in their wall, so they have some constrictive ability, but they're much thinner, the veins, than the arteries. If you ever look on a cadaver and you're looking at the arteries and the veins, you know, you've seen probably, if you've done that at all, a lot of you probably haven't, but the arteries and the veins and the nerves in the periphery tend to run together. They run those three structures together. So if you're looking at any particular one, you'll often see the arteries and the veins and the nerves. Well, the arteries, if you feel them, they have a thicker wall, and the veins have a thin wall, and the nerves have no lumen. They're completely solid. Okay? So a lot of times that's one way to tell them apart, because they're not nicely color-coded on a cadaver or in our bodies as they are in the diagrams. So the venous system is going to hold on to a lot of our blood volume. The veins act as a reservoir for a lot of our blood volume. And it slowly moves back to the heart. In the arteries, the blood is moving quite rapidly, and in the veins, it's moving quite slowly. And of course, venous return is very important, because if we don't get the blood returned to our heart, it eventually will affect our end-diastolic volume and our cardiac output. So we do need venous return, but it just returns more slowly. Well, how does the blood return to the heart? What is the factors that affect venous return? Well, one factor is there is some pressure in the veins. It's not near as much as in the arteries, but we still have some pressure and some pressure gradients in the veins. That's one factor, even though it's much less of a factor than in the arteries. Another factor is when we take a breath in, inspiration in our thoracic cavity increases in volume, it decreases in pressure, according to Boyle's law. It's an inverse relationship at constant temperature. Now you might say, what? Say that again. Boyle's law simply states that at constant temperature, pressure and volume or space are inversely related. So if you increase your volume or space your thoracic cavity, it decreases the pressure. What makes that important here is that that pressure is now lower than the pressure in our veins, and so that draws the blood back to the heart. So that's number 2. And number 3 is the skeletal muscle pump. Oh, what's that all about? Arteries, veins, and nerves often run through skeletal muscles, in between muscles and that kind of thing. Yeah, we have muscles all over the body. When these muscles contract, because the veins are so thin-walled, it compresses the veins and squishes them and forces the blood back towards the heart. Now, why doesn't it force it away from the heart? Because we have one-way valves. And so the veins, unlike the arteries, have one-way valves. So they open in the direction going back to the heart, and then they close so that it doesn't go backwards. And so this propels the blood back to the heart, these one-way valves. And so that's called the skeletal muscle pump. Here it is. And this shows the skeletal muscles contracting. Here's our one-way valves, and it prohibits it from going backwards, but it allows it to go back towards the heart. Now, we know that venous return is important. If you line a bunch of people up and put them at attention, especially if they're in the sun, after about an hour or less, they'll start fainting. What's happening is, if you have them at attention, you've prevented them from contracting their skeletal muscle and forcing that blood back toward the heart. So it's pooling. Now you're standing at attention, so gravity is also pulling it down to the lower extremities. If it's the hot sun, it makes it worse because now you're sweating and you're trying to shunt blood to your periphery, your skin, and that kind of thing to cool you off. And after a while, you're not getting enough end-diastolic volume, you're not getting enough cardiac output, you're not getting enough blood and oxygen to your brain. Your brain says, I'm the boss. If I'm in trouble, we're all in trouble. Push the red button and you faint. And then that allows the blood flow to start going to your brain again. And then you revive. And so we need that venous return. Another bad thing is if we don't move the blood from the veins as promptly and efficiently as we should, the blood can start to clot. This is why if you take a long flight to Japan or Australia or somewhere, you need to get up or move around a bit. Don't just sit there, because your blood will tend to cool down. And if you're not moving, you're not getting those skeletal muscle things contracting, and you're not getting that back up. So we don't want to stay too stationary if you're doing a long trip. You need to have that for a venous return. This is why if you're in the choir and they say, oh, this year for Christmas we're going to sing the Messiah, and we want to look sharp, so we're going to put you in robes. We want everybody to see you, so we're going to put some nice spotlights on you. And you're standing there and you're singing this long piece. You could faint. You need to do a little knee bends, discreetly under there, and you need to maybe have a little water you're sipping and all those kind of things, because you could actually, you could cause your choir members to faint if you're putting them in that tough physiological situation for too long. Okay, so that's kind of venous return, and that's how that works. But what about blood flow in general? We've used this equation for blood flow, and this could be either arteries or veins. We use it probably more for arteries, but it could be for either one. And this is our equation. Delta P is our change in pressure. You recognize delta from math classes. Delta P is our change in pressure. Change in pressure from where to where? From the beginning of the blood vessel to the end of the blood vessel. That's our delta P. R in this case stands for radius of the blood vessel. And notice it's to the 4th power. It's an exponent. So that's a very important one because it's to the 4th power. And then we have a constant pi, 3.14, etc. Okay? And that's just part of the constant of the equation. So the delta P and the radius are going to be have a direct relationship. If you increase these, you increase the blood flow. And then it's underneath, you have the length of what? The blood vessel. So that's inverse related. And then mu, Greek letter, that stands for viscosity here, okay? That's viscosity, how thick the blood is. So that's inverse related. The thicker it is, the harder, it is to flow. And then 8 is also a constant in the equation. So you can see that the delta P is important, the radius is very important, and the length has some importance. And viscosity. So if we're looking at this, notice here, if everything else is equal, the change in pressure, it's the change in pressure, not the total pressure that's important. This is illustrated by, here in our top example, the pressure is 100 millimeters of mercury at the beginning and 10 millimeters of mercury at the end of the vessel. So our delta P is 90. In our example 2, it's 500 at the beginning and 410 at the beginning. It would have the same flow rate. It would be delta P is 90 if everything else is equal. So it's our change in pressure, not our overall pressure. That's important there. Now what about the radius? Here we see that the radius of this opening is a unit of 2, and the radius of this one over here is 1. In other words, this is twice the size. This opening is twice the size of this. Well, it doesn't just double the flow rate. How much would it increase it? How much is this greater, the flow rate, than this? Everything equal except for the radii. It'd be 16, wouldn't it? Why? Because it's to the 4th exponent. 2 times 2, 4, 4 times 2 is 8, 8 times 2 is 16, and so just doubling your radius will give you a 16-fold change in your flow rate, assuming everything else like your delta P and viscosity are equal. So that tells you how important that is. Now in blood vessels, that's our vasoconstriction and our vasodilation. We can change the diameter, or in this equation it's the radius, half the diameter, but same idea, right? We can change how big the openings are for our blood vessels. We can make them bigger or smaller, and this is especially true, of course, of our arteries, our muscular arteries and our arterioles. And so we can change the flow rate, and part of the reason that's helpful is, again, like we've mentioned, we can move blood flow around to different parts of the body that's needed, and also we can control our blood pressure by changing that. And so that shows how important the radius is, and that it is changeable. Well, the last bit, we'll talk about blood pressure. Blood pressure, we've talked about a little bit. We've said that in general we use the term 120 millimeters of mercury for our systolic blood pressure and 80 for our diastolic, and that's our resting blood pressure. If we exercise, of course, it goes up much more. You did a little bit of that in lab. Well, you were looking at blood pressure though, but did you look at blood pressure? Anyway, you did a little bit of that exercise and increasing things. But basically, we know that when we exercise, our heart rate goes up and our blood pressure goes up, and this is perfectly normal. Then it has to go back down again. One challenge is we can get hypertension, and that's just our blood pressure is too high. Our resting blood pressure is really what we're looking at. And although we get different numbers from different sources, usually we say anything above 140 systolic or above 90 diastolic, we would say is hypertension. Some people would put it a little lower, some people would put it a little higher, but that's about a pretty good marker for us. Well, how do we control our blood pressure? We know it has to go up when we exercise and come back down after we exercise. We have some baroreceptors. These are pressure receptors. They respond to pressure. These baroreceptors are in some of our larger arteries. They're in the aorta, the aortic arch. They're in the carotid. We have a common carotid, and it divides into an internal and external carotid. These arteries go up into our neck, and we have these baroreceptors there as well. Well, these pressure receptors are constantly monitoring the pressure of the blood in the blood vessels. And if it's too low, we will generally have sympathetic norepinephrine, epinephrine as a hormone, that will increase our heart rate, increase our blood pressure, and we'll have vasoconstriction, also increases our blood pressure a bit. This is controlled by our vasomotor center in our medulla oblongata. It's one of the vital reflex areas. If you want to come back to physiology, you said enough person. Anyway, that increases it, right? Okay. That is controlled by our vasomotor. It means talking about blood pressure that's going on. What about if it's too low, too high, then we do our parasympathetic. We decrease our sympathetic and we increase our parasympathetic. And we decrease our heart rate, we have vasodilation, and we bring it back into balance. Chemoreceptor. We have baroreceptor that help control this. We also have chemoreceptors in the periphery. They're in the aorta and the carotid arteries. And then we have some central in the brain. These are looking at oxygen content, carbon dioxide, and hydrogen ion, or pH. We're going to review these again in the next chapter with respiratory. But essentially, let's just say if you have a decrease in oxygen or an increase in carbon dioxide and increase in hydrogen ion, which is a decrease in pH, any of these things will lead to an increase in blood pressure by that same mechanism. If, on the other hand, you have an increased O2, a decreased CO2, a decreased hydrogen ion, you can either raise or lower your blood pressure depending on these factors and the pressure given. So that's the two. Hypertension we've been talking about. Hypertension is too high of a resting blood pressure. It can cause a lot of problems. Heart failure over time. Remember we talked about the afterload, so every time you're trying to pump blood out of the heart, your stroke volume, you have a resistance, your blood pressure, your afterload, and if you increase that too much all the time, it's okay while you're exercising, but if you increase it all the time, day and night, every week, you can wear out the heart. And so hypertension can lead to heart failure over time. Renal failure. It puts too much pressure and it starts destroying the glomerular capillaries in the kidneys. That's where you filter blood from the blood into the renal tubules, and if it's too high a pressure over time, you can break down that filtration barrier. Stroke, it can cause a leakage or a problem in the brain. You have a case study that's a little bit related to that, hypertension, which is a risk factor for stroke. And then, of course, it damages your endothelial lining of your blood vessels and can lead to vascular diseases. So these are some of the problems. Most hypertension does not have a single cause, and this is called primary or essential hypertension. This is a type of hypertension that we may not even know why all the reasons, but secondary, primary is about 90%, secondary is only about 10%. Secondary means you know exactly what's causing it. Like you have a tumor in the adrenal medulla, so you have too much epinephrine, and it's causing you to have hypertension. Or Cushing's disease, again, you can have too high of an output, this time from the adrenal cortex, and so you get these problems. So there's different types, but most of it is primary or essential, which is multifaceted and not really, don't necessarily know all of the reasons. How can it develop and how can we sort of treat it? For one thing, if we have a high sodium chloride intake, this affects certain types of hypertension more than others, salt related, but it can be a problem. High cholesterol, that lowers the elasticity of the arteries if you have a high cholesterol, because it gets inserted, forms plaques in your arteries. Obesity, you have to pump blood to a greater amount of tissue. Age, that's because our arteries wear out after a while, and as we get older, they lose their elasticity. They become more like hardened pipes instead of elastic pipes, and that can cause hypertension. Gender, males get it more than females, probably because estrogen has some protective role, and then after menopause, the rates for women go up to approach that of men. So we believe that's probably due to protective effects of estrogen, and estrogen falls, then there's that. Stress certainly can be a factor. Diabetes mellitus, because it damages the endothelium, the higher sugar, and that causes the arteries again not to be as pliable, flexible. It can run in families. The salt-sensitive forms are more prevalent in black people than white people, so there is some problem. I mean, that's some genetics evidence as well. Stress, smoking is very damaging. Smoking is so bad for everything, right? So smoking is, chemicals in smoking also can increase the blood pressure. We try to manage it with diet, exercise, and lifestyle changes. If those don't work, we resort to medications, and a lot of people are on hypertension medications as they get older. Diuretics make you pee more, you know that. A lot of the first medication that somebody with high blood pressure is going to go on is a diuretic. You might say, well, that's kind of weird, but it doesn't have as many side effects as some of the others. And so you're trying to decrease the blood volume so it decreases the pressure. Now you can't get too carried away, you could overdo it, but within limits, a diuretic can be very helpful. Beta blockers, that blocks the heart contractility and heart rate a bit, calcium channel blockers, they do the same thing. ACE inhibitors, we'll look at that on the next slide, but that's an angiotensin converting enzyme inhibitor. And angiotensin 1 antagonist is something similar. Well, let's look at these last two. Do you remember seeing this in the endocrine system a bit, or? Yeah, so this is the same thing. One of the hormones that raises our blood pressure is aldosterone. It's not the only one that epinephrine and norepinephrine does, norepinephrine as a neurotransmitter does, but aldosterone reabsorbs more sodium ions and water from our kidney nephrons. So it allows less sodium and water to go into the urine, and it grabs it back for our blood. And that raises our blood pressure. So if our blood pressure goes low, we want to secrete more aldosterone. But it's a several-step process. If our blood pressure goes low, we have a precursor protein that's already in the blood called angiotensinogen. And angiotensinogen is made by the liver. And so it's just like a lot of proteins. It's a protein, and it's there. And if our blood pressure is dropping, of course we're going to release epinephrine, but we're going to release renin from the kidneys. And renin is an enzyme that's going to convert angiotensinogen to angiotensin 1. Well, then angiotensin 1 in turn is going to be converted by another enzyme, angiotensin 2. Now this enzyme is called angiotensin converting enzyme. It's mostly made by endothelial cells, maybe a little bit by the lungs as well. Angiotensin converting enzyme, what do we call it for short? ACE, angiotensin converting enzyme. A lot of people that are on blood pressure medicines to decrease it are on ACE inhibitors, right? It's inhibiting this enzyme right there. So it's inhibiting this step. And so that's what it is. And so you don't make as much angiotensin 2. Angiotensin 2 is a mild vasoconstrictor on its own, so you inhibit that. But you also, angiotensin 2 stimulates aldosterone secretion from the zona glomerulosa of the adrenal cortex. So you reduce your aldosterone. So you can block angiotensin 2. You can also block angiotensin 1. You block their receptors. That's this AT1. So they can't bind to cause the release of angiotensin 2. Antidiuretic hormone. If you have more antidiuretic hormone, you would increase your blood volume and your blood pressure. A diuretic is the opposite of that. It causes you to pee more, and so that's where a diuretic would fit in. You could theoretically block antidiuretic hormone as well if in a person with high blood pressure. So just by understanding the background physiology there, then we can understand how some of these blood pressure medications work. What questions do you have, or what would you like to chat about before we leave this chapter? Feel okay about it? Some of the stuff in here is new. All right, well, let's go to the respiratory chapter. We'll just get a decent start on it today, and then we'll be continuing on the rest of the week. The respiratory chapter really ties in very nicely with the cardiovascular chapter, because they really rely on each other a lot. When we look at the respiratory system, we're looking at these four main processes. Pulmonary ventilation is breathing, okay? External respiration is getting oxygen from the lungs into the blood, and carbon dioxide from the blood into the lungs. Then we have transport of these respiratory gases, oxygen and carbon dioxide, in the blood. And finally, we have internal respiration, the movement of oxygen from the blood to the tissue cells, and CO2 in the opposite direction, so that oxygen can be used as the final electron acceptor of our electron transport chain for cellular respiration. We know that oxygen is necessary as the final electron acceptor, and then it turns into water. So our byproducts of cellular respiration, we get ATP, energy, yay, and then we get water, and we get carbon dioxide, okay? We always know what to do with water. You know, if you have too much, you pee it out, otherwise you use it for some other purpose. Carbon dioxide, though, we have to get it back into the blood, take it to the heart, back to the lungs, and breathe it out. This is also important, not only just to get rid of the carbon dioxide, but to maintain our pH. Because the more CO2 we have, the more hydrogen ions we have, the more acidic our blood or our internal environment becomes. So respiration and breathing have an impact on our pH as well. Here's this little movement where oxygen is coming in and moving, and carbon dioxide is going the other way. Interestingly enough, the movement of oxygen and carbon dioxide is by simple diffusion. It goes from a higher concentration of partial pressure, we often use that term in respiratory, to a lower concentration of partial pressure. We'll get into partial pressures and what they are quite a bit more, probably tomorrow. So here's the functions of the respiratory system. This is from your book. The two first ones are the primary, providing oxygen and eliminating carbon dioxide. Gas exchange. Then we have, as we mentioned, regulation of pH. Speech, well, that's a pretty important one too. Defending us against microbes. Well, the respiratory system is very vulnerable to pathogenic organisms because it has a very large surface area. You know, we need an exchange between the lung tissue, like the alveoli, and the blood, and the capillaries, the pulmonary capillaries, if you will. Now, if you took, one lung, and you took a big rolling pin, and you flattened out the lung tissue and the blood capillaries, and you made it this nice, big, huge pancake, it would be about the size of a tennis court. It would be about 75 square meters. That's a huge surface area. It's great for oxygen and carbon dioxide exchange. We need it to be huge, but it's a vulnerability for the body because now pathogens and stuff can get in there. So we have a number of protective things. We have some dust cells or kind of macrophages, phagocytic cells, white blood cells in our lungs that help eat up the bacteria. But our respiratory tract, like our trachea and bronchi, it's lined with a type of epithelium called pseudostratified ciliated columnar epithelium. Okay, well let's not worry about that so much, but we have little mucus cells in there called goblet cells, and the mucus gets secreted, and the mucus is sticky. So dust particles and bacteria and all that sticks in the mucus in our respiratory tract. Well, we don't want it to stay there. The cilia move it, the mucus with all this junk in it, away from our lungs, up to our throat, our pharynx, and then we swallow it, and the acidic environment of the stomach takes care of it. And so we have this constant staircase movement of this mucus and all the junk in it to keep it out of our lungs. And we also have the coughing reflex and all that. Smokers, one of the bad things is their cilia get temporarily paralyzed with smoking, and so it's not moving and stuff out. And so especially in the morning, you get the smoker's cough. You're trying to get all that junk out of your lungs because the cilia are not moving it out for you as they would properly want to do. We make a few chemical messengers in the lungs, and we also trap and dissolve blood clots from the legs. They come up to that pulmonary system, and the pressure is lower in there, and they're able to get rid of blood clots. Now, if you have a huge blood clot, maybe after surgery or something, you can have a pulmonary embolism that is so big that it gets caught and gets fatal. So you can have a really bad, huge clot. But if it's a smaller clot, then the pulmonary circulation in your lungs helps get rid of the clot. If it's really big, it's a pulmonary embolism like PE. Well, ventilation is breathing. We need to breathe in and out. We're taking oxygen in from the atmosphere, and we're breathing out carbon dioxide. When we look down at our alveoli, an alveolus is the singular for one of these little air units. Alveolus is singular, alveoli is plural. This shows a macrophage, a dust cell. This shows some of the pulmonary capillaries with the red blood cells in there. The structure of the alveoli are two types of cells, type 1 and type 2. Type 1 are structural cells. They make up the structure of the alveolus. Type 2 are also there, but they produce surfactant. Surfactant helps expand the lungs. We'll get back to that one too. That's two types. This is the name of surfactant right here. Surfactant right there. But we'll get back to that and its function a little more. This shows you the airway, the nasal cavity are coming through the mouth, you go through the pharynx, then you come down through the trachea. The trachea, of course, is protected by these cartilaginous rings that we can feel so that our airway doesn't collapse. Our larynx is where our voice box is. We have our vocal cords in there that can vibrate as air goes across them. And then we have a branching. We go to the bronchi, and then the bronchioles, and they branch and branch and branch as we go further in the lungs. Here's the airways that we see. As we go beyond the larynx, we can divide the airways into a conducting zone and a respiratory zone. What's the difference? Conducting zone is where you're taking air in and using that energy, but it never gets to a part of the lungs where you have exchange of gases. No oxygen and carbon dioxide. So you have to move the air in and move the air out, but you don't get any benefit directly from it. The respiratory zone is that volume of air that we inhale that reaches the gas exchange zones, oxygen and carbon dioxide. And so they're called the respiratory zones or regions. These really start at the respiratory bronchioles and then they go the rest of the way. This is your conducting zone. Again, we have the larynx with some cartilage around it, and we have the trachea with these cartilaginous rings, and then we have the bronchi. We talked about the mucus and the cilia that is within this region for protection. we finally get to our last structure of the pathway. These are the alveoli, or the alveoli. And we have about 300 million alveoli in each lung. The surface area we mentioned briefly, that's 75 square meters, about the size of a tennis court, and 300 million alveoli. Each alveolus is only one cell thick, and the total barrier between the blood, pulmonary capillary, and the air in the alveoli is about 2 microns. Anything that increases that diffusion barrier decreases our efficiency of gas exchange. Pneumonia, where you get fluid in the lungs, congestive heart failure, where you get fluid in the lungs, are just some examples. Here's our alveolar type 1 cells, make structure. Alveolar type 2 secrete surfactant. This is the branching. Here's our conducting zone, and then here's our respiratory zone. Our respiratory zone is respiratory bronchioles, alveolar ducts, and alveolar sacs. Alveolar sacs are not the same as alveoli. Alveolar sacs are like clusters of grapes. If you picture a grape cluster, each individual grape is an alveolus. The cluster is an alveolar sac. We have about 8 million alveolar sacs, grape clusters, and we have about 300 million alveoli, individual alveoli. So lots of those that we see. The conducting zone, it needs to be a low resistance pathway. It defends us against microbes and chemicals and stuff, with cilia and mucus and macrophages. It warms and moistens the air and helps with speaking vocal cords. Here's our respiratory bronchiole. This is an example of an alveolar sac. You see how it kind of has that grape cluster look. And there's about 8 million of these per lung. And then if you count up all these individual alveoli, like grapes, there's about 300 million of those. In all of this region, we have gas exchange. It's exchanging oxygen and carbon dioxide with the blood. Prior to this, before that, that's our conducting zone. The air comes in and out, but there's no gas exchange. This shows you that the pulmonary capillaries wrap around the alveoli, and this gives us this huge surface area. Here's our pulmonary capillaries. Notice that the pulmonary arteries are carrying deoxygenated blood coming from the right side of the heart, and then they go through the pulmonary capillaries, and our pulmonary veins carry oxygenated, 4 pulmonary veins, and that carries oxygenated blood back to the left atrium. Again, very thin wall between, about 2 microns, so we get lots of good gas exchange under normal circumstances. Setting up for ventilation, breathing, we have some structures around the lungs called the pleura. Pleura are double-layered membranes. We have pleura in 3 main locations. We have these double-layered membranes in 3 main locations. The pleura is the double-layered membrane around our lungs, the pleura. The pericardium is the double-layered membrane around our heart, and the peritoneum is the double-layered membrane around many of our abdominal organs. So the pleura is the first of these. The pleura, as well as the pericardium and peritoneum, have 2 layers. The inner layer is called the visceral layer, and the outer layer is called the parietal layer. The visceral layer actually is the outer layer of the lungs. And then you have just this small little potential space, and then you have the parietal pleura. There's a little film of fluid between the 2 that help them move against each other with lubrication. The parietal pleura is really attached to our inner thoracic wall. And the viscera is our outer part of the lung. Here is the parietal pleura, and here is our visceral pleura. This is our interpleural fluid and potential space. If you imagine putting your fist in a fluid-filled balloon, the inner layer here would be the visceral pleura, this would be your lungs, the fist, and this would be the outer layer would be your parietal pleura. We expand the lungs during breathing by stretching out on the parietal pleura, which causes a little space in between, and the visceral pleura moves into that space. And it's based on the fact of the force of air is the atmospheric pressure minus the alveolar pressure. The atmospheric pressure is the pressure in the atmosphere, here, barometric pressure, if you will. The pressure in the alveoli is our lung pressure inside the lung. And then divided by resistance to airflow. That's the force, or that's our airflow equation. Air enters the lungs when the pressure in the alveoli in the lungs is less than the atmospheric pressure. Air exits the lungs when the pressure in the alveoli is greater than the pressure in your atmosphere. It's really as simple as that. We move it down a pressure gradient, and that's how we do it. So, since we mentioned Boyle's law, we'll end on this. Boyle's law is simply stated, at a constant temperature, the volume, space, is inversely related to pressure. So if we decrease the volume, we'll increase the pressure. And if we increase the volume, we'll decrease the pressure, excuse me. They're inversely related. And so that simple principle is what determines our breathing in and out. And that's really where we're going to hit it. Tomorrow we'll start